385 million years ago, something happened on the surface of the Earth that had never happened before in the 4 billion-year history of the planet. A tree grew. Not a small plant. Not a ground-hugging moss or a low fern. A tree, a tall, woody organism with a thick trunk, deep roots, spreading branches, and a canopy of leaves that cast shade on the ground below. The first tree on Earth rose above a landscape that had never seen shade, that had never experienced the fall of a large quantity of leaves, that had never had deep woody roots penetrating the soil to the depths of rock beneath, and that had never had to process the death of something so large and so chemically complex. The emergence of forests in the Devonian period, between approximately 385 and 360 million years ago, was one of the most consequential events in the history of life on Earth. It changed the chemistry of the atmosphere. It changed the temperature of the planet. It changed the soil, the rivers, and the oceans. And it may have triggered the late Devonian mass extinction, the second of the 5 great mass extinctions in the history of complex life, which eliminated approximately 75% of marine species and transformed the biological world in ways that are still felt today. Subscribe now and join the channel. Every week a new documentary, always calm, always going somewhere unexpected, always in the hours designed for thinking and for rest. The story of the first forests is one of the most extraordinary stories in the history of life, and it is one that most people have never heard, because the organisms at the center of it are not the charismatic animals that dominate popular accounts of deep time. Plants are slower than animals. Their dramas unfold over millions of years rather than individual lifetimes. But the drama of the first forests is as significant as any in the history of life, and it involves some of the most revolutionary biological innovations ever produced by evolution. To understand what made the first forests revolutionary, it is necessary to understand what the world looked like before they appeared. In the early Devonian period, approximately 400 million years ago, the land surface of the Earth was largely bare. The continents that existed at that time, assembled into configurations quite different from those of today, were covered in low-growing vegetation. Mosses, liverworts, simple vascular plants with no true leaves, and the first primitive land plants that had only recently made the transition from aquatic to terrestrial life. Those plants were small, rarely reaching more than a meter in height, and they had shallow root systems that barely penetrated the surface of the mineral soil. The landscape they inhabited would have looked, from a distance, something like a modern tundra or a rocky moorland, green in places, barren in others, with no tall vegetation to break the horizon. The time between the appearance of the first vascular land plants in the Ordovician period, approximately 470 million years ago, and the appearance of the first trees in the Middle Devonian, approximately 385 million years ago, was 85 million years of slow, incremental evolution during which land plants gradually developed the structural and physiological capabilities needed to grow tall. That 85 million year period of preparation included the development of the first true vascular tissues, the water-conducting xylem and the sugar-conducting phloem that allow plants to move water and nutrients between roots and leaves over distances greater than a few centimeters. It included the development of true roots capable of penetrating into the soil rather than simply anchoring the plant at the surface. And it included the development of the first true leaves, photosynthetic organs that dramatically increased the surface area available for capturing sunlight. Each of those innovations was a prerequisite for the appearance of the first trees, and each took millions of years to develop through the normal mechanisms of natural selection acting on genetic variation within plant populations. The transition from that low-growing vegetation to the tall woody forests of the late Devonian was one of the most rapid and far-reaching evolutionary innovations in the history of land plants, and it required the development of several biological innovations that had never previously existed. The most fundamental was wood. Wood, or more precisely secondary xylem, the rigid cellulose and lignin framework that supports the stems and trunks of woody plants, is the biological material that makes tall trees possible. Without wood, a plant cannot grow tall without collapsing under its own weight, because the structural limits of the soft tissues of herbaceous plants prevent them from reaching more than a few meters in height. Wood provides the combination of rigidity and strength needed to support a tall columnar structure against the force of gravity and the buffeting of wind, and it also serves as the primary conducting tissue that transports water from the roots to the leaves over distances of tens of meters. The paleoecology of Devonian stream and river environments adjacent to the first forests reveals an ecosystem of extraordinary complexity and novelty. The rivers of the Devonian world were not simply channels of water flowing to the sea. They were biological processing units that transformed the chemical composition of the water passing through them, removing nutrients from the river water by biological uptake and adding others through decomposition of organic matter falling from the banks. The first forests dramatically increased the input of organic matter to those river systems, as leaves and branches and eventually entire fallen trees entered the waterways. The accumulation of wood in Devonian and Carboniferous river channels is well documented in the geological record. Log jams and wood-rich deposits are found in many late Devonian and Carboniferous river sediments, and some of those wood-rich deposits have been preserved as the coal seams that underlie much of Europe and North America. The chemical composition of wood, particularly the high proportion of lignin, the complex polymer that stiffens the cell walls and makes them resistant to decomposition, was also one of the key factors in the environmental consequences of the first forests. Lignin had never existed in significant quantities in the terrestrial environment before the appearance of woody plants, and the organisms capable of decomposing it, the fungi and bacteria that today break down fallen wood and return its carbon to the atmosphere, had not yet evolved the enzymatic machinery needed to break the lignin polymer efficiently. The result was that when the first trees died and fell, their wood did not decompose rapidly, but instead accumulated in massive quantities on the forest floor and in the rivers and lakes of the Devonian world. That accumulation of undecomposed wood has a visible legacy in the modern world. The coal seams of the Carboniferous period, which were laid down in the 300 million years following the first appearance of forests, are composed primarily of the compressed and carbonized remains of those first forest trees, preserved because the organisms capable of decomposing them had not yet evolved. The Industrial Revolution that drove the development of the modern world was powered by the solar energy captured by those Devonian and Carboniferous forests, stored in their wood for 300 million years and released when the coal was burned. The first trees inadvertently stored the fuel of the Industrial Age. The deep root systems of the first forest trees were another biological innovation of the first importance. The roots of Devonian forest trees, like those of modern trees, penetrated the soil to depths of meters rather than the centimeters achieved by the root systems of the early land plants. Those deep roots broke up the bedrock, creating channels through which water could flow deep into the soil, and they produced organic acids that accelerated the chemical weathering of the silicate minerals in the rock. That enhanced weathering had a profound effect on the global carbon cycle. The chemical weathering of silicate rocks is one of the primary mechanisms by which carbon dioxide is removed from the atmosphere over geological timescales, and the dramatic expansion of deep-rooted vegetation in the Devonian accelerated that removal to rates far higher than anything the Earth had previously experienced. The first trees were not the tall, straight-trunked conifers or broad-leaved trees that define most people's mental image of a forest. They were stranger, more experimental, and in some ways more extraordinary than anything that grows on Earth today. The best known of the first forest trees is Archaeopteris, which appears in the fossil record approximately 370 million years ago and is often described as the world's first modern tree in the sense that it had both woody tissue similar to that of modern trees and true leaves. Archaeopteris was a large tree by any standard. Specimens reaching 30 meters in height and trunk diameters of more than a meter have been found in the fossil record. Its leaves were arranged in frond-like branches that spread horizontally to capture sunlight, and its reproductive strategy involved the production of spores rather than seeds, making it technically a fern-like plant despite its tree-like overall form. Archaeopteryx forests covered large areas of the late Devonian tropical and subtropical world, forming dense canopies that shaded the ground below and transforming the terrestrial ecosystem in ways that can still be read in the chemical composition of the rocks deposited at that time. The Gilboa fossil site in New York State, where some of the most important early tree fossils have been found, was itself the product of a specific geological circumstance that preserved materials that would otherwise have been lost. The stumps at Gilboa were preserved by a rapid volcanic ash fall that buried the base of a standing forest approximately 385 million years ago. The ash infiltrated the spaces between the roots and hardened around the basal portions of the trunks, preserving their three-dimensional form in extraordinary detail, while the upper portions of the trees rotted away after the ash buried the base. When the ash deposit was subsequently buried under river sediment and eventually exposed by erosion, the preserved stumps were there, waiting for the eyes of a 19th-century scientist who could recognize their significance. The Gilboa Quarry, which has been actively worked for stone since the 19th century, has been an ongoing source of new botanical discoveries as each new face of rock is exposed. But Archaeopteryx was not the first tree. That distinction belongs to a group called the Cladoxylopsids, which appeared approximately 385 million years ago in the early part of the Middle Devonian. The Cladoxylopsids are among the most biologically peculiar organisms ever discovered, and understanding how they worked required the physical sectioning and microscopic analysis of fossil trunks preserved in 3 dimensions in ancient volcanic deposits. The best-studied Cladoxylopsid is Watiza, specimens of which were found in upstate New York in 2004 in a quarry near the town of Gilboa. The Gilboa fossils had been known since the 19th century, but the discovery of 3-dimensionally preserved trunks in 2004 made it possible to analyze the internal anatomy of Watiza in detail for the first time. What the analysis revealed was a tree that worked in a way that is entirely unlike any living tree. Instead of a central column of wood with rings of annual growth, the trunk of Watiza consisted of multiple strands of woody tissue running separately through a core of spongy non-woody material, like a bundle of tubes embedded in foam. That architecture, which has no modern equivalent, allowed the tree to grow tall without the solid central trunk that characterizes all modern trees, and it worked. Watiza reached heights of 8 to 10 meters in a landscape that had previously seen no vegetation taller than half a meter. The diversity of solutions to the problem of growing tall that the first forests explored is one of the most striking features of Devonian forest ecology. Multiple independent lineages of plants evolved woody tissues and tall growth forms within a relatively short period of geological time, each using a different structural solution to the engineering problem of supporting a large plant against gravity. That convergent evolution of height and woodiness, with different lineages arriving at different structural architectures, reflects the powerful selective advantage of being tall in the newly forested Devonian world. Access to more sunlight, the ability to overtop competitors, and the dispersal advantage of releasing spores from a greater height. The ecological transformation produced by the first forests was not limited to the land. The effects of the spread of Devonian forests propagated into the rivers, the lakes, and ultimately the ocean, changing those environments in ways that had profound consequences for the organisms living in them. The coevolution of the first terrestrial vertebrates with the first forests is one of the most fascinating stories in the history of life, and it involves organisms that are among the most popular subjects of paleontological research. The first tetrapods, the 4-limbed vertebrates that descended from lobe-finned fish during the late Devonian, were initially amphibious animals that spent most of their time in the water but were capable of venturing onto land for periods of time. The environments that those early tetrapods inhabited were the swampy, vegetated margins of Devonian rivers and lakes, precisely the environments that were being transformed by the spread of the first forests. The leaf litter, fallen wood, and dense root systems of the first forests created microhabitats on the forest floor that were accessible to small amphibious animals and that were rich in the invertebrate food that those animals depended on. The forests did not just transform the atmosphere and the oceans. They created the specific ecological niches that allowed the first tetrapods to become more fully terrestrial. The deep roots of Devonian forest trees broke up the bedrock and created new pathways for water flow through the soil. That increased water penetration changed the hydrology of the landscape. Rivers that had previously flowed across relatively impermeable surfaces began to receive more water from deep soil infiltration, and the rivers themselves changed character. The geomorphological evidence from late Devonian river deposits shows a transition from the braided river systems that characterized the pre-forest landscape, characterized by multiple shallow channels separated by gravel bars, to meandering river systems with single deeper channels. A transition driven by the increased cohesion of riverbanks due to plant root binding and by the changed timing of water delivery to the rivers from the newly forested catchments. The chemical composition of river water also changed dramatically with the spread of forests. The enhanced weathering of silicate rocks by the acid-producing roots of the first trees released large quantities of silica, calcium, magnesium, potassium, and phosphorus into the rivers, which carried those nutrients into the coastal ocean. Phosphorus is a particularly important nutrient for aquatic ecosystems, because it limits the productivity of freshwater and marine algae in most environments, and the large pulse of phosphorus delivered to the Devonian Ocean by the first forests triggered blooms of algae and cyanobacteria of extraordinary intensity. Those algal blooms had direct consequences for the oxygen content of the water. When the algae died and sank to the seafloor, the bacteria that decomposed them consumed oxygen faster than it could be replenished from the atmosphere. The result was the development of widespread oxygen-depleted zones in the Devonian Ocean, particularly in the shallow shelf environments where most marine life was concentrated. The black shales that characterize many late Devonian geological sections are the preserved sediments of those anoxic environments. Fine-grained sediments deposited in oxygen-free conditions that prevented the burrowing activities of bottom-dwelling organisms and preserved organic matter in extraordinary quantities. The extinction of the Devonian forest trees and the transition to the coal swamp forests of the Carboniferous was not a simple replacement of one flora by another, but a complex ecological transition that involved the successive decline of some plant groups, the diversification of others, and the appearance of entirely new evolutionary lineages that had not previously existed. The late Devonian mass extinction that eliminated the majority of marine species also affected terrestrial plant communities, and the early Carboniferous saw the replacement of the Devonian forest composition by a different assemblage of plants in which the lycopods, the horsetail relatives, and the early seed plants became dominant at the expense of the Devonian tree ferns and the cladoxylopsids. That transition took millions of years and was not uniform across all parts of the world. Different continental regions made the transition at different rates and through different intermediate stages, reflecting the geographical variation in climate and ecology that characterized the late Devonian and early Carboniferous world. The relationship between the spread of Devonian forests, the enhanced weathering of rocks, the delivery of phosphorus to the ocean, the algal blooms, the development of ocean anoxia, and the late Devonian mass extinction is now one of the best documented examples of how a biological innovation on land can propagate through multiple Earth systems and produce environmental changes of global scale. The first trees did not intend to trigger a mass extinction. They were simply doing what plants do: photosynthesizing, growing, reproducing, and dying. But the cumulative effect of millions of trees doing those things simultaneously, in a world that had no experience of those things and no adaptations to cope with their consequences, was a cascade of environmental changes that eliminated the majority of marine species living at the time. The carbon dioxide drawdown associated with the spread of the first forests was one of the most significant perturbations to the atmospheric greenhouse in the history of the Earth, and its effects on global climate were as dramatic as anything produced by any subsequent geological or biological event. Before the appearance of forests, the concentration of carbon dioxide in the atmosphere was approximately 10 to 15 times higher than today's levels, maintained by the balance between volcanic outgassing and the relatively slow chemical weathering of silicate rocks by the low-growing early land plants. The photosynthesis of those early plants removed carbon dioxide from the atmosphere, but the rapid decomposition of their simple organic tissues returned most of that carbon to the atmosphere within years of the plant's death, creating a relatively short carbon cycle that did not significantly affect the atmospheric carbon dioxide concentration. The appearance of forests changed that balance in two ways simultaneously. First, the photosynthesis of the tall, large-leafed Devonian trees was significantly more efficient per unit of ground area than that of the low-growing plants they replaced, removing more carbon dioxide from the atmosphere per year. Second, and more importantly, the lignin-rich wood of those trees resisted decomposition, allowing carbon to be stored in the terrestrial environment for decades, centuries, or millennia rather than years. That storage of carbon in long-lived woody tissues and in accumulating undecomposed wood created a new carbon reservoir on land that had not previously existed, drawing down atmospheric carbon dioxide at a rate that the ocean carbon cycle and the volcanic outgassing of carbon could not compensate for. The geochemical evidence for this carbon dioxide drawdown is preserved in multiple independent archives. The isotopic composition of the carbon in organic matter from late Devonian sediments shows the signature of greatly increased biological productivity, consistent with enhanced photosynthesis by the spreading forests. The weathering proxies in late Devonian soils show a dramatic increase in the depth and intensity of chemical weathering, consistent with the enhanced acid production of deep tree roots. And the Paleosol record, the preserved ancient soils of the late Devonian, shows the development of deep, organically rich soil profiles that had not previously existed, consistent with the accumulation of undecomposed organic matter under the first forests. The result of all that carbon drawdown was a significant cooling of the global climate. The late Devonian was a period of global cooling that has been identified in both the marine and terrestrial fossil records, with evidence for the advance of glaciation at high latitudes. That cooling is the third mechanism, alongside ocean anoxia and the nutrient fertilization of the ocean, by which the spread of the first forests contributed to the late Devonian mass extinction. Cold conditions reduced the biodiversity of the shallow marine environments where most Devonian life was concentrated, eliminating tropical and subtropical species that could not tolerate the lower temperatures. The study of fossilized pollen and spores from Devonian sediments has provided a different and complementary perspective on the spread of the first forests from that provided by the macrofossils of leaves, wood, and trunks. Pollen and spores are produced in enormous quantities by plants and are dispersed widely by wind and water before settling in sediments, where they can be preserved for hundreds of millions of years in the chemically resistant outer wall of their spore cases. The analysis of those microscopic fossils, known as palynology, allows the reconstruction of the plant communities that existed at a site even when the plants themselves have left no macroscopic remains. The Devonian palynological record shows the progressive appearance and spread of different plant types, including the characteristic spores of Archaeopteris and other late Devonian trees, and it has been used to establish the global biogeography of Devonian vegetation in far more detail than would be possible from macrofossil data alone. The fossil forests of the Devonian period have been found in several locations around the world, and the most spectacular of those deposits are themselves the products of the kind of geological accidents that preserve the ephemeral in the permanent. The role of fungi in the early history of forests is far more central than their relative obscurity in popular accounts of the Devonian would suggest. Fungi were not passive inhabitants of the first forests but active participants in the ecological processes that defined those forests. The mycorrhizal fungi that colonized the roots of the first trees extended the effective absorptive surface of those roots by orders of magnitude, allowing the trees to access nutrients that would otherwise have been unavailable to them in the nutrient-poor soils of the early land environment. Without mycorrhizal assistance, the first trees might not have been able to grow on the mineral soils that were all that was available before the first forests had created the organic soils that make forest growth self-sustaining. The relationship between the first trees and their fungal partners was therefore not a luxury but a necessity, and it was one of the earliest and most successful ecological partnerships in the history of terrestrial life. The Gilboa Fossil Forest in New York State, the site of the Watiza discoveries, was formed by a rapid volcanic ash fall that buried the base of a standing forest approximately 385 million years ago. The ash preserved the stumps of the trees in growth position, allowing scientists to reconstruct the three-dimensional structure of the forest floor. The spacing of the trees, the density of the roots, and the composition of the ground vegetation between the trunks. The Gilboa forest was a sparse, open forest more similar to a palm grove or a bamboo stand than to the dense closed canopy forests of the modern tropics, with individual trees spaced several meters apart and a ground layer of ferns and other plants between them. The Devonian fossil forest at Schönbrunn in Germany, which was discovered in a quarry in 2021 and dates to approximately 360 million years ago, near the end of the Devonian period, preserved an entire forest floor in 3 dimensions. The fossils, embedded in the rock of a former river floodplain, include the trunks, root systems, and fallen branches of multiple species of Devonian trees, providing a view of late Devonian forest composition and structure that had not previously been available from any other site. The forest contained Archaeopteris trees alongside at least 2 other types of trees, suggesting that by the late Devonian, the composition of forests was already diversified beyond the simple pioneer forests of the early Devonian. The fossil forest at Joggins in Nova Scotia, Canada, while dating from the subsequent Carboniferous period rather than the Devonian, preserves the most complete picture of early forest ecosystems available anywhere in the geological record. The Joggins fossil cliffs, which are now a UNESCO World Heritage Site, expose a sequence of Carboniferous sediments containing hundreds of fossil tree stumps in growth position, along with the remains of the plants and animals that lived between the trees. The stumps of the giant lycopod trees, Sigillaria and Lepidodendron, which dominated the Carboniferous coal swamp forests, stand up to a meter in diameter and represent trees that reached heights of 40 to 50 meters. Within the hollow stumps, the bones of early reptiles and amphibians have been found, animals that had taken shelter in the cavities left when the inner tissues of the stumps rotted away, and that became trapped when the stumps filled with sediment during periodic floods. Those fossil forests are windows into a world that was simultaneously alien and foundational. Alien because the organisms that composed them were unlike anything alive today, and foundational because the ecological processes they initiated, the accumulation of organic matter, the weathering of rock, the transformation of soil, and the cycling of carbon through the terrestrial environment, are the same processes that underlie the functioning of all subsequent forests, including those of today. The organisms that lived in and around the first Devonian forests were as strange and as unprecedented as the forests themselves. The Devonian period is often called the Age of Fishes, and with good reason. The late Devonian saw the most spectacular diversification of fish in the history of the vertebrate lineage, including the appearance of the first fish to breathe air using a primitive lung-like organ, the first fish to develop limb-like fins capable of supporting their weight on land, and the first vertebrates to actually walk on land rather than simply crawling briefly out of the water. Those evolutionary transitions, which would eventually produce the entire lineage of terrestrial vertebrates, including human beings, were intimately connected with the appearance of the first forests. The shallow rivers and coastal swamps of the late Devonian, bordered by the first forests, were environments of fluctuating oxygen levels, where the seasonal decomposition of fallen leaves and wood in warm water could temporarily deplete the dissolved oxygen to levels that made gill breathing impossible. The fish that survived in those environments were those with the ability to breathe air directly, supplementing their gill respiration with a primitive lung or using the swim bladder as an air-breathing organ. That ability, which was the key innovation required for the subsequent colonization of the land, evolved in response to the specific conditions created by the first forests. The biogeography of the first forests reflects the continental configuration of the Devonian world, which was significantly different from the modern arrangement of continents. In the early Devonian, the continents were distributed across a range of latitudes, with the southern supercontinent Gondwana extending from the South Pole to the tropics and the northern continent Laurussia, which included what would become North America and northern Europe, occupying subtropical and tropical latitudes. The first forests appeared in the tropical and subtropical regions, where the warm, wet climate was most favorable for the growth of the spore-reproducing plants that dominated the first tree flora. As the Devonian progressed, forests spread into higher latitudes, following the retreating ice of the late Devonian glaciation and colonizing the temperate zones that had previously supported only low-growing vegetation. By the end of the Devonian, forests were present at latitudes as high as those of modern Scotland and Scandinavia, though the trees of those high-latitude forests were smaller and less diverse than those of the tropical forests. The discovery in 2019 of a Devonian fossil forest in Antarctica, in rocks exposed by erosion near the South Pole, demonstrates that forests reached even the high southern latitudes during the late Devonian, in a period when the Antarctic continent was not ice-covered but supported a temperate climate that allowed trees to grow. The Antarctic fossil trees, which represent a high-latitude equivalent of the Archaeopteris forests of the lower latitudes, grew in conditions of extreme seasonality, with months of continuous winter darkness and months of continuous summer light. The growth rings in the fossil would show clear annual increments, demonstrating that the trees had a seasonal growth rhythm adapted to the extreme light regime of the polar environment. That adaptation, which required the ability to survive months of darkness in a dormant state before resuming growth in the polar summer, prefigures the deciduous habit that would evolve in Carboniferous and Permian trees as the climate became more seasonal. Tiktaalik rosea, discovered in the Canadian Arctic in 2004, is the most famous representative of the transitional organisms that bridged the gap between fish and the first land-walking vertebrates. It had fins that contained a recognizable precursor of the arm and hand bones of tetrapods, including a wrist-like joint that would have allowed it to prop itself up on its fins, and it had a neck, which fish do not have. It lived approximately 375 million years ago, at the height of the Devonian forest expansion, in a river environment bordered by Devonian forest trees. The appearance of Tiktaalik and similar transitional organisms was not a coincidence of timing. The spread of the first forests created the specific environmental conditions of shallow, oxygen-depleted, vegetated water that made the evolution of air-breathing and eventual land locomotion advantageous. The arthropods of the Devonian forests were among the first truly terrestrial animals in the history of life. Myriapods, the ancient relatives of millipedes and centipedes, were abundant in Devonian forest ecosystems, feeding on the decaying plant material that accumulated on the forest floor. Some of those Devonian myriapods reached extraordinary sizes. Arthropleura, a giant millipede-like arthropod that appeared in the Carboniferous period and represents the culmination of the trend that began with Devonian arthropods, reached lengths of up to 2.5 meters, making it the largest land invertebrate that has ever existed. Those giant arthropods were possible only in the oxygen-rich atmosphere that the photosynthesis of the first forests helped to create. The end of the Devonian period, approximately 360 million years ago, was marked by the late Devonian mass extinction, which eliminated approximately 75% of marine species and transformed the terrestrial fauna in ways that are still being studied. The marine casualties of the late Devonian extinction included the stromatoporoids, sponge-like organisms that had been the primary reef builders of the Silurian and early Devonian, and that were eliminated so completely that reef ecosystems did not recover for approximately 20 million years after the extinction. The placoderms, the armored fish that had been the dominant vertebrates of the Devonian Ocean, were entirely eliminated, along with many groups of corals, trilobites, and other marine invertebrates. The terrestrial record of the late Devonian extinction is less complete, but it shows significant changes in plant communities and the disappearance of some early tetrapod lineages. The role of the first forests in triggering the late Devonian extinction has been recognized for decades, but the specific mechanisms by which the forests contributed to the extinction have been debated and are still not fully resolved. The current consensus, based on the integrated analysis of the geochemical, sedimentological, and paleontological records of the late Devonian, is that the forests contributed through at least three mechanisms: the drawdown of atmospheric carbon dioxide, which cooled the climate; the enhanced delivery of nutrients to the ocean, which triggered algal blooms and ocean anoxia; and the increased erosion and sediment delivery to the coastal ocean, which disrupted shallow marine habitats. The irony of the late Devonian extinction is that it was in large part a consequence of the most important biological innovation in the history of terrestrial life: the appearance of forests. The trees did not intend to cause an extinction. They were following the same evolutionary logic that drives all living things: to grow, to reproduce, to spread into available space. But the cumulative effect of millions of trees doing those things simultaneously, in a world that had evolved without trees for the preceding 4 billion years, was an environmental perturbation of global scale. Life had never before produced a single innovation with such far-reaching consequences for the chemistry of the atmosphere, the hydrology of the continents, and the ecology of the oceans. That pattern, in which a biological innovation of extraordinary success creates environmental conditions that are lethal to many of the organisms living at the time, is a recurring theme in the history of life. The Great Oxidation Event of approximately 2.4 billion years ago, in which photosynthetic cyanobacteria flooded the atmosphere with oxygen that was toxic to the anaerobic organisms that had dominated the Earth for billions of years, is the most dramatic earlier example. The late Devonian forest extinction is its terrestrial equivalent, the triumph of one evolutionary innovation creating environmental conditions that were catastrophic for others. The evolution of bark is a biological story as extraordinary as the evolution of wood, though it receives far less attention in popular accounts of plant evolution. Bark is not simply a protective covering for the wood beneath. It is a complex, metabolically active tissue that performs multiple essential functions for the tree, including protection against physical damage, pathogens, and herbivores, regulation of the movement of gases and water between the interior of the tree and the atmosphere, and in some trees the storage of nutrients and water. The first bark appeared in the Devonian period as woody plants evolved the need to protect their water-conducting tissues from drying out and from the organisms that would exploit them. The diversity of bark types in modern trees reflects the different evolutionary histories of different plant lineages and the different ecological pressures they have faced, but all of them share the fundamental architecture of the first bark: a protective outer layer of dead cells underlain by a living inner bark through which sugars are transported from the leaves to the roots. The recovery of life after the late Devonian extinction produced the Carboniferous period, the age of coal forests, which in some ways represents the full flowering of the biological potential that the first Devonian trees had introduced. The Carboniferous coal swamp forests were dominated by giant lycopods, tree-like plants related to the humble club mosses of today, but reaching heights of 40 to 50 meters with trunk diameters of more than a meter. Those trees, which reproduced by releasing spores rather than seeds, grew in the vast tropical swamp forests that covered much of the equatorial region of the late Carboniferous world, when the continents were assembled into the supercontinent Pangaea and the equatorial regions experienced a warm, humid climate that supported the development of the densest forests the Earth has ever seen. The chemical signals preserved in Devonian black shales provide some of the most direct evidence for the anoxic ocean conditions that accompanied the spread of the first forests and contributed to the late Devonian mass extinction. Black shales, which are sedimentary rocks rich in organic matter and depleted in oxygen, form in environments where the rate of organic matter deposition exceeds the rate at which it can be decomposed by aerobic organisms. The black shales of the late Devonian are found on every continent and show a consistent geochemical signature of anoxic conditions: the presence of framboidal pyrite, which forms only in sulfidic anoxic environments, the depletion of molybdenum, which is removed from the water column under reducing conditions, and the elevated concentrations of organic carbon, which accumulates when decomposition by aerobic organisms is prevented by the absence of oxygen. Those chemical signals tell the story of an ocean suffocating under the nutrient load delivered by the first forests. The structural biology of the Carboniferous lycopods is as extraordinary as that of the Devonian cladoxylopsids. Instead of growing a solid column of wood like a modern tree, the lycopods grew a hollow cylinder of bark-like tissue around a central core of soft tissue, like a living hollow tube. The bark performed the structural role that would plays in modern trees, but it was produced by a completely different mechanism. The tree shed its lower leaves and branches as it grew taller, and the leaf scars left on the bark surface, which created the distinctive diamond-shaped pattern visible in fossil lycopod bark, thickened and hardened into the structural tissue that supported the tree. The coal that powers a significant fraction of the world's electricity today, that was the fuel of the Industrial Revolution, and that built the material foundation of modern civilization, is the compressed remains of those Carboniferous lycopod forests. The coal seams of Pennsylvania, Wales, the Ruhr Valley, and Siberia are the remnants of forests that existed 300 million years ago, forests whose wood accumulated in vast quantities because the organisms capable of decomposing lignin-rich tissue had still not fully evolved. The carbon stored in those trees, captured from the atmosphere by photosynthesis during the Carboniferous period, was locked in the rock for 300 million years and is now being released back into the atmosphere by human combustion, at a rate incomparably faster than the rate at which it was originally captured. That connection between the first forests of the Devonian period, the coal forests of the Carboniferous, and the climate crisis of the 21st century is one of the most direct links between deep geological time and the immediate human present. The carbon dioxide that is warming the planet today was removed from the atmosphere by the first forests and their successors between 300 and 400 million years ago. Its release by industrial combustion is returning the atmosphere to conditions not seen since before those forests existed. The first trees changed the world by removing carbon from the atmosphere. Human civilization is changing the world by putting it back. The seed, which appears in the fossil record approximately 365 million years ago, at approximately the same time as the late Devonian extinction, was the botanical innovation that would eventually allow the forests to reclaim the world after the extinction and to spread into environments that the spore-reproducing plants of the Devonian could not colonize. The reproductive strategy of the first forest trees, like that of modern ferns, involved the release of spores that could only develop into the next generation of the plant if they landed in a moist environment. Spores have no nutrient reserves and no protective coat. They are essentially naked cells that must germinate immediately or die. That limitation confined the spore-reproducing plants of the Devonian to environments with reliable moisture, leaving large areas of the continental interior, far from the ocean and experiencing significant seasonal drought, inaccessible to the first forests. The climate of the early Devonian world, before the appearance of forests, was significantly warmer and wetter than today at most latitudes, and the high carbon dioxide concentration of the atmosphere maintained a strong greenhouse effect. The geological evidence for that warm climate includes the widespread distribution of coral reefs at latitudes that today are too cold to support tropical coral growth, the presence of evaporite deposits indicating warm, shallow seas in what are now cool continental interiors, and the absence of any evidence for polar ice caps. That warm, wet climate was one of the factors that made the Devonian such a favorable time for the explosive diversification of fish in the world's oceans. Warm, nutrient-rich seas supported the productivity that sustained the extraordinary diversity of Devonian marine life. But it was also the climate that the first forests began to change, by drawing down the carbon dioxide that maintained the warmth and cooling the planet toward the glacial conditions that would characterize the subsequent Carboniferous and Permian periods. The seed transformed that limitation by providing the developing plant embryo with a protected nutrient-rich package that could survive drying, remain dormant for months or years, and germinate when conditions became favorable. A seed is not just a reproductive unit but a survival mechanism, allowing plants to colonize environments that would be lethal to a vulnerable seedling if it germinated at the wrong time. The appearance of the first seed plants, The pteridosperms or seed ferns, in the late Devonian and early Carboniferous, opened the continental interiors to plant colonization for the first time and set the stage for the eventual spread of forests across the entire terrestrial surface of the Earth. The subsequent evolution of the gymnosperm and angiosperm lineages from those first seed plants produced the most diverse and most ecologically dominant group of organisms in the history of the terrestrial world. The conifers, cycads, ginkgos, and flowering plants that dominate terrestrial ecosystems today are all descended from those first seed plants of the late Devonian and early Carboniferous, all sharing the fundamental innovation of the seed that their ancestors developed during the ecological turmoil of the late Devonian mass extinction. The timing is significant. The seed appears precisely at the time when the Devonian world was undergoing the most severe environmental stress of its history. The innovation that would eventually allow forests to cover the world was produced by the same catastrophe that was simultaneously eliminating most of the organisms that the first forests had displaced. The first forests also transformed the soil in ways that had consequences for all subsequent terrestrial life, including the soil ecosystems that underlie all modern agriculture. Before the first trees, the soil of the terrestrial environment was a relatively thin, poorly developed layer of weathered mineral material with little organic content. The early land plants had begun the process of soil formation, but their shallow roots and rapid decomposition did not produce the deep, organically rich soils that support most modern terrestrial life. The deep roots of the first Devonian forest trees penetrated the soil to depths of meters, physically breaking up the rock below and chemically weathering it with the organic acids they produced. The leaves and wood that fell from those trees accumulated on the forest floor and were slowly decomposed by the first soil organisms, creating the organic horizon that lies at the top of modern forest soils. The research into the first forests has also illuminated the origins of plant secondary metabolism, the production of complex organic molecules that plants use for defense against herbivores and pathogens. The terpenes, phenolics, alkaloids, and other secondary metabolites that give plants their characteristic scents, tastes, and medicinal properties are produced by biochemical pathways that evolved in response to the ecological pressures of the terrestrial environment, including attack by herbivorous insects and fungal pathogens. Many of the secondary metabolic pathways found in modern plants have their origins in the Devonian period, when the first land plants were developing their chemical defenses against the first land herbivores. The lignin that makes wood resistant to decomposition is itself a form of secondary metabolite, produced by a biochemical pathway that evolved as a structural material but that also functions as a chemical defense against herbivores and pathogens. The first forests were not just physical structures. They were chemical landscapes, permeated by the complex organic molecules of plant secondary metabolism. That organic horizon, the layer of partially decomposed organic matter that lies between the surface of the soil and the mineral substrate below, is the foundation of terrestrial productivity. It is where most of the nutrient cycling in terrestrial ecosystems takes place, where the bacteria and fungi that decompose organic matter release the nitrogen, phosphorus, and potassium that plants need, and where the mycorrhizal fungi that form symbiotic associations with the roots of most modern plants live and work. Without that organic horizon, the nutrient cycling that supports the growth of plants and the animals that eat them would be far less efficient, and the productivity of terrestrial ecosystems would be a fraction of what it currently is. The symbiotic relationship between the first forest trees and mycorrhizal fungi is one of the most ancient and most important ecological partnerships in the history of life. Mycorrhizal fungi, which colonize the roots of plants and extend the effective root surface area of the plant by orders of magnitude, are not a recent innovation. They are documented in the fossil record of early land plants dating back more than 400 million years, and their appearance in the fossil record coincides with the earliest successful colonization of the land by plants. The first forest trees probably depended on mycorrhizal fungi for the acquisition of phosphorus and other nutrients in the same way that modern forest trees do, and the spread of those fungal networks through the soil of the first forests created a biological internet that connected individual trees to each other and allowed the transfer of nutrients and signaling compounds between them. That interconnected underground network of fungal filaments, which the popular science writer Merlin Sheldrake has called the Wood Wide Web, was already operating in Devonian forests. The trees of those ancient forests were not isolated individuals competing for resources, but members of a connected community, sharing resources and information through the soil network in ways that we are still learning to understand. The discovery that this ecological partnership predates the appearance of the first forests themselves, and that it may have been essential for the success of the colonization of the land by complex plants, is one of the most significant findings in the recent history of paleobotany. The Devonian period takes its name from the county of Devon in southwestern England, where the rocks of that age were first studied systematically in the 1830s by the British geologists Roderick Murchison and Adam Sedgwick. The naming of geological periods after the regions where their type sequences are exposed is a practice that dates from the early 19th century, when geologists were first working out the stratigraphy of the rock record in Britain and Europe. That historical circumstance means that many of the most important geological periods, including the Devonian, the Cambrian, the Ordovician, and the Silurian, are named after places in Wales and England, reflecting the priority of British geology in developing the stratigraphic framework of the Phanerozoic Eon. The Devonian rocks of Devon itself are not among the most important or most complete exposures of that period. Far richer sequences are exposed in the Rhine Valley of Germany, in the Ardennes of Belgium and France, in the Canadian Arctic, and in eastern North America. But the name Devon has been attached to the period for nearly 2 centuries, and it will continue to be used regardless of where the most significant Devonian research is conducted. The chemical revolution in the atmosphere produced by the first forests is one of the most dramatic examples of how life shapes its own environment over geological timescales. Before the appearance of forests, the oxygen content of the atmosphere was lower than it is today, and the balance between photosynthesis and decomposition was such that the net flux of oxygen into the atmosphere from biological processes was relatively small. The appearance of forests changed that balance dramatically. The large-scale storage of carbon in wood and in undecomposed organic matter in the first forest soils and swamps removed carbon from the biological cycle and sequestered it in long-lived organic deposits, creating a net flux of oxygen into the atmosphere from the excess photosynthesis that could not be balanced by decomposition. The result was a dramatic rise in atmospheric oxygen over the Carboniferous period, following the initial carbon dioxide drawdown of the Devonian. Atmospheric oxygen in the late Carboniferous reached approximately 35%, compared with the current 21%, the highest oxygen concentration in the atmosphere since the origin of complex multicellular life. That high oxygen concentration had profound effects on the terrestrial fauna of the Carboniferous. Insects, which diffuse oxygen directly through their exoskeletons rather than using lungs or gills, could grow to sizes impossible at lower oxygen concentrations, producing the giant dragonfly-like Meganeura and the enormous Arthropleura. And the risk of wildfire, which increases dramatically with atmospheric oxygen concentration, was also elevated. The Carboniferous fossil record contains abundant evidence of charcoal from forest fires, indicating that wildfires were a regular feature of Carboniferous forest ecosystems in a way that they are not in modern forests with lower atmospheric oxygen. The atmospheric oxygen content eventually stabilized as the deposition of organic carbon in the coal swamps slowed and the evolution of wood-decomposing fungi in the Permian period finally brought the decomposition rate of lignin-rich tissues closer to the rate of their production. The Permian period saw the decline of the great coal swamp forests and the spread of drier, more open woodlands, and the atmospheric oxygen concentration gradually fell back toward its current level as the decomposition of the accumulated organic deposits by the newly evolved wood-rotting fungi released the stored carbon back to the atmosphere as carbon dioxide. That biological revolution in decomposition, The evolutionary development of the white rot fungi that produce the enzymes capable of breaking the lignin polymer is one of the most consequential evolutionary events in the history of the terrestrial biosphere, and it represents the closing act of the drama that began with the appearance of the first forests. The legacy of the first forests in the modern world extends beyond the coal that fuels our energy system and the soil that feeds our agriculture. The evolution of deciduousness, the habit of shedding leaves in autumn and regrowing them in spring, is an adaptation that appeared in the Carboniferous and Permian periods in response to the increasingly seasonal climate of the drying continental interior of Pangaea. The deciduous habit allows trees to avoid the metabolic cost of maintaining leaves through seasons when light or water is limiting, and it also produces the spectacular display of autumn color that is one of the most distinctive features of the temperate forest landscapes of the modern world. That display, which is caused by the breakdown of the green chlorophyll in the leaves as the trees withdraw nutrients from them before shedding, was first seen in forests that grew on the margins of the drying Permian deserts, hundreds of millions of years before any human being was present to appreciate it. The debate among paleobotanists about the phylogenetic relationships of the first forest trees, the question of which modern plant groups are most closely related to the Devonian tree lineages, has been transformed by the application of molecular phylogenetics to living plants and by the detailed morphological analysis of fossil material using synchrotron imaging and other new techniques. Those analyses have confirmed that the Devonian Cladoxylopsids, including Watiza, are the earliest members of the fern lineage rather than the ancestors of the seed plants as some earlier researchers had proposed. Archaeopteris, which for decades was considered a candidate ancestor of the seed plants because of its leaf structure, has been reassigned to a separate extinct lineage with no living descendants. And the seed plants themselves, which eventually came to dominate all terrestrial ecosystems, are now known to have diverged from their non-seed plant relatives in the Middle Devonian, approximately 380 million years ago, well before the late Devonian extinction that eliminated the Cladoxylopsids and many other early tree lineages. The evolution of the flowering plants, the angiosperms, in the Cretaceous period, approximately 130 million years ago, represents the most recent major revolution in the history of forests, and it produced the most diverse and most ecologically complex forests in the history of the Earth. The angiosperms, with their co-evolutionary relationships with pollinators, seed dispersers, and herbivores, created ecological networks of a complexity that far exceeds anything that preceded them. The tropical rainforests that represent the modern expression of that angiosperm diversity are the most species-rich terrestrial ecosystems on Earth, and they are descended, through a lineage of geological transformations that includes the first Devonian forests, from the earliest land plants that colonized the bare rock of the Ordovician and Silurian continents. The fossil record of the first land insects appears in the Devonian period, coincident with the appearance of the first forests, and the relationship between the two is almost certainly not a coincidence. The earliest definitive insect fossils, dating to approximately 400 million years ago in the early Devonian, precede the appearance of forests by approximately 15 million years, but the diversification of insect lineages accelerates dramatically in the Middle and Late Devonian and the early Carboniferous, in parallel with the spread of forests. The first forests provided insects with an entirely new ecological opportunity: the exploitation of plant tissues as food, using wood, bark, leaves, roots, and other plant parts as resources that had not previously been available to terrestrial invertebrates. The evidence for plant-insect interactions in Devonian fossil plants includes the distinctive damage patterns produced by herbivorous insects on leaves and stems, which can be identified in fossil plant material from the late Devonian onward. Those interactions, which represent the earliest documented examples of herbivory in the terrestrial world, were the beginning of the co-evolutionary arms race between plants and plant-eating animals that has been one of the primary drivers of terrestrial biodiversity for the past 350 million years. That lineage, from the first land plant to the first forest tree to the first seed plant to the first flowering plant to the modern tropical rainforest, is one of the longest and most consequential evolutionary trajectories in the history of life. The organisms at the beginning of that trajectory would be unrecognizable to anyone familiar only with modern forests, strange, experimental, structurally novel organisms that were solving problems of terrestrial existence for the first time and producing solutions that had never been tried before. But they were the founders of all that followed, the originators of the biological processes and the ecological relationships that make the terrestrial world habitable for the extraordinary diversity of life that exists today. The research into the first forests has been transformed in recent years by new analytical techniques that allow scientists to extract information from ancient plant material that was previously inaccessible. The analysis of fossil wood using synchrotron X-ray tomography, which can reveal the three-dimensional structure of cells in a fossil without destroying the specimen, has made it possible to study the anatomy of Devonian and Carboniferous wood in a detail that was impossible with conventional microscopy. Those studies have revealed the diversity of wood anatomies in the first forests, with different tree lineages using different structural strategies for supporting tall growth, and have provided new information about the evolutionary relationships between those early wood types and the wood of modern trees. The stable isotope analysis of fossil plant material has provided information about the atmospheric conditions in which the first forests grew, including the carbon dioxide concentration of the atmosphere and the temperature and precipitation at the sites where the plants grew. The carbon isotope composition of fossil organic matter reflects the atmospheric carbon dioxide concentration at the time of its formation through the process of carbon isotope discrimination during photosynthesis, and the oxygen isotope composition of fossil wood cellulose reflects the temperature and humidity of the environment in which the tree grew. Those isotopic records have been used to reconstruct the atmospheric and climatic conditions of the Devonian period with a precision that was not previously achievable. The molecular analysis of ancient organic molecules preserved in Devonian and Carboniferous sediments is providing a new kind of window into the chemistry of the first forests. Certain organic molecules, particularly the hopanoids produced by bacteria and the steranes derived from the cell membranes of eukaryotic organisms, are preserved in ancient sediments for hundreds of millions of years and can be used to identify the types of organisms that were living in a particular environment at a particular time. The analysis of those molecules in Devonian sediments has revealed the presence of specific groups of bacteria that are associated with the decomposition of organic matter under anoxic conditions, consistent with the development of the oxygen-depleted conditions that triggered the late Devonian marine extinction. The paleobotanical research that is reconstructing the first forests is conducted in some of the most remote and least accessible parts of the world, because the Devonian and Carboniferous rocks that contain the most significant plant fossils are often exposed in locations that require arduous travel to reach. The fossil forests of the Canadian Arctic, which contain some of the most complete Devonian plant assemblages in the world, are accessible only by helicopter or by foot across tundra terrain. The Devonian beds of Greenland, which have produced some of the most important early tetrapod fossils, are exposed in cliff sections that can only be reached by boat in the brief Arctic summer. The fossils of the first forests are as inaccessible as the forests themselves were different from anything in the modern world. Tonight, as you prepare for sleep, the first forests of the Devonian are present in the world around you in ways that are invisible but fundamental. The significance of the first forests for understanding the current climate crisis is one of the most practically important insights that paleobotany can offer. The late Devonian carbon dioxide drawdown, produced by the appearance of forests, provides a natural experiment in the effects of large-scale changes in the terrestrial carbon cycle on the global climate. The rate of carbon dioxide removal from the atmosphere by the first forests, while impressive by geological standards, was far slower than the current rate of carbon dioxide addition by human combustion. The geological timescale over which the first forests changed the climate, millions of years, is incomparably longer than the timescale over which human emissions are changing the climate, decades to centuries. But the directional effects are the same. Changes in the terrestrial carbon cycle, driven by biological processes, can produce significant changes in atmospheric carbon dioxide concentration and global temperature. The first forests changed the world by removing carbon from the atmosphere. Human industrial activity is changing it by putting carbon back. The experience of standing in a modern forest and trying to imagine what the world looked like before forests existed is an exercise that requires a significant effort of deconstructive imagination. Everything about the forest environment that we take for granted, the shade, the leaf litter, the deep humus soil, the smell of decomposing wood, the sound of wind in the canopy, the mycorrhizal network connecting the roots below, and the diverse community of organisms from insects to birds to mammals that the forest supports, was absent from the pre-forest world. The earliest land surfaces were bare mineral rock or thin mineral soils, exposed to the full force of wind and rain and sunlight without the moderating influence of vegetation. The rivers were clearer, carrying less organic matter and fewer nutrients. The atmosphere was different in composition. And the silence of the pre-forest world, without the biological sounds produced by the forest community, would have been broken only by wind and water. The first trees did not simply grow in that world. They created a new world, one that has been the foundation of all subsequent terrestrial life. The oxygen you are breathing was produced, in its modern atmospheric proportions, largely by the photosynthesis of the first forests and their descendants over the 350 million years since the Devonian. The soil beneath any garden or field is the product of the soil-building processes that the first forest trees initiated, the accumulation of organic matter, the weathering of rock by deep roots, and the development of the mycorrhizal networks that connect plant roots to the nutrients they need. And the coal that still powers a significant fraction of the world's electricity was the wood of those first forests, compressed and carbonized over 300 million years of geological time. The trees outside your window, if there are trees, are the descendants of a lineage that began in the Devonian period. Their wood is made by the same biochemical processes that made the wood of Archaeopteryx and Watiza. Their roots interact with mycorrhizal fungi in the same way that the roots of Devonian forest trees did. Their leaves capture sunlight by the same photosynthetic process. And when they die and fall, their wood is decomposed by the white rot fungi that evolved specifically to break down lignin, the same fungi whose ancestors ended the era of coal formation by learning to break down the polymer that the first forest trees had invented. The first forests were not just a geological curiosity, an episode in deep time that has no relevance to the present. They were the foundation of the terrestrial world that makes the present possible. Without the first forests, there would be no deep soil to grow food in, no oxygen-rich atmosphere to breathe, no coal or oil to power the industrial world, no lineage of woody plants from which the timber, the paper, the medicines, and the ecosystems that we depend on are derived. The world before trees was a world inhospitable to the kind of terrestrial life that eventually produced us. The trees changed that. They changed it by accident, by being what they were and doing what plants do, but they changed it permanently and completely. Sleep now, beneath the oxygen that the first forests helped to create, in a world of deep soil and tall trees and the quiet biological processes that have been running for 385 million years. The first forests are still here, transformed beyond recognition but continuous in their chemistry and their ecology with the trees that started it all in the Devonian. They are the most ancient achievement of terrestrial life, and the most enduring. That continuity, from the first tree to the forest outside your window, is one of the longest unbroken threads in the history of life on Earth. It stretches across 385 million years, through every major extinction and every major climate change, maintained by the same fundamental biological processes that the first Devonian trees initiated. Rest with that thought. The question of how the first trees grew so tall so quickly in evolutionary terms, achieving heights of 10 meters or more within a few million years of the first appearance of woody tissue in the fossil record, has been addressed by recent biomechanical modeling studies that demonstrate the structural advantages of different wood architectures under specific growth conditions. Those models show that the bundle architecture of Watiza and the other cladoxylopsids, while fundamentally different from the central wood column of modern trees, was mechanically efficient for rapid vertical growth in the specific conditions of the early Devonian. The bundle architecture allowed the tree to grow tall using less metabolic energy than a solid wood column would have required, at the cost of lower structural resilience in high wind conditions. That trade-off between growth rate and mechanical resilience, which favored rapid height growth in the relatively sheltered valley environments where the first forests grew, explains why the bundle architecture was successful in the early Devonian despite its structural limitations. The nitrogen cycle was profoundly altered by the appearance of the first forests, with consequences that rippled through both terrestrial and marine ecosystems. Nitrogen is an essential element for all life, and its cycling between the atmosphere, the soil, and living organisms is one of the most important biogeochemical processes on Earth. Before the first forests, the nitrogen cycle in terrestrial environments was limited by the scarcity of organic matter in the soil, which restricted the activity of the nitrogen-fixing bacteria that convert atmospheric nitrogen into the forms usable by plants. The deep organic soils created by the first forests provided the conditions for a greatly enhanced nitrogen cycle, with more nitrogen-fixing bacteria, more nitrifying bacteria, and more denitrifying bacteria all active in the organic horizon. That enhanced nitrogen cycling increased the availability of usable nitrogen in terrestrial ecosystems, supporting higher plant productivity and more diverse plant communities. But it also increased the delivery of nitrogen to rivers and eventually to the ocean, contributing to the nutrient enrichment and algal blooms that drove the late Devonian marine extinctions. The relationship between the first forests and the evolution of flight in insects is one of the more indirect but fascinating consequences of the appearance of tall vegetation on land. The first winged insects appear in the fossil record in the early Carboniferous period, approximately 320 million years ago, approximately 50 to 60 million years after the appearance of the first forests. The evolutionary pressure for the development of insect flight is thought to have been related to the colonization of the new ecological opportunities offered by the first tall plants, the ability to disperse from plant to plant without descending to the ground, to access the pollen and spores produced at the tops of tall plants, and to escape from ground-dwelling predators by taking to the air. Without the first forests, there would have been no selective pressure for the development of insect flight, and without insect flight, the subsequent coevolutionary history between flying insects and flowering plants, which produced much of the biological diversity of the modern world, would not have occurred. The Rhynie Chert of Scotland, a silica-rich rock deposit dating to approximately 407 million years ago, is one of the most important fossil sites in the history of paleobotany, though it predates the first forests by approximately 22 million years. The Rhynie Chert preserves an early land plant community in extraordinary three-dimensional detail, including the cells, vascular tissues, and reproductive structures of multiple plant species, the first land arthropods, and the earliest known fungi living in symbiosis with plant roots. The Rhynie community did not include any trees. The plants preserved in the chert were small, simple vascular plants no more than a few centimeters tall. But the mycorrhizal fungi preserved in association with those early plant roots demonstrate that the partnership between plants and fungi that was essential for the development of the first forests was already established more than 20 million years before the first tree grew, providing the biological foundation on which the forest ecosystem would eventually be built. The late Carboniferous and early Permian saw the gradual drying of the coal swamp environments that had supported the great lycopod forests, as the climate of the interior of the supercontinent Pangaea became more seasonal and less humid. That drying, which eliminated the permanently waterlogged conditions that the lycopods required for reproduction by spore, drove the transition from the lycopod-dominated coal forests to the conifer-dominated and seed fern-dominated forests of the late Permian. The conifers, with their needle-like leaves that conserve water, their woody cones, and their drought-resistant seeds, were better adapted to the drier Permian climate than the lycopods, and they diversified rapidly as the lycopods declined. By the end of the Permian period, The great coal forests were gone, replaced by forests of a character more similar to the modern boreal forests of Siberia and Canada. That transition, which took approximately 50 million years, was the final act of the first forest revolution, the replacement of the biologically experimental forests of the Carboniferous by the more familiar, more modern forest types that would persist, in modified form, to the present day. The contribution of paleobotany to the understanding of climate change is one of the least recognized but most significant contributions of any branch of paleontology to contemporary science. The fossil plant record provides direct evidence for the atmospheric carbon dioxide concentrations and temperatures of past periods, evidence that cannot be obtained from any other source and that is essential for calibrating the climate models used to project future change. The stomatal density of fossil leaves, the number of breathing pores per unit area of leaf surface, varies systematically with the carbon dioxide concentration of the atmosphere. Leaves that grew in high carbon dioxide atmospheres have fewer stomata per unit area than leaves that grew in low carbon dioxide atmospheres, because fewer stomata are needed to obtain sufficient carbon dioxide for photosynthesis when the atmosphere is rich in it. Measuring the stomatal density of fossil leaves from different periods provides a direct measure of the atmospheric carbon dioxide concentration at the time the leaves were alive, and the measurements from Devonian and Carboniferous leaves have confirmed the dramatic drawdown of carbon dioxide that accompanied the spread of the first forests. The geography of Devonian continental positions created specific conditions for the development of the first forests that would not have existed in a different paleogeographic configuration. The tropical continental landmasses of the Middle Devonian provided the warm, wet environments in which woody plants could develop most rapidly, while the higher latitude landmasses provided the seasonal environments that drove the evolution of deciduousness and the other adaptations that allowed forests to spread into less hospitable climates. The shallow epicontinental seas that covered much of the lower latitude continental interiors in the Devonian provided the vast areas of shallow marine habitat that supported the diverse marine fauna that would be eliminated by the late Devonian extinction. And the continental configuration of the Devonian, with the major landmasses distributed across a wide range of latitudes without a single large polar continent to anchor a global circulation pattern, may have made the climate particularly sensitive to the changes in greenhouse gas concentrations produced by the spread of the first forests. The concept of forest as a distinct ecosystem type, with its own characteristic climate, hydrology, species composition, and ecological dynamics, is one that was implicit in the appearance of the first Devonian forests, but that took hundreds of millions of years to fully develop into the complex, self-regulating systems that characterize modern old-growth forests. The first Devonian forests were ecologically simple compared with modern forests. They lacked most of the animal diversity that contributes to the ecological complexity of modern forests. They had less diverse plant communities than modern tropical or temperate forests, and the soil ecosystems that underlie modern forest productivity were only beginning to develop. But they contained the essential elements that define a forest: tall woody plants that modify the local climate by creating shade and reducing wind, deep root systems that transform the soil chemistry and hydrology, and an annual input of organic matter from leaf fall and wood decomposition that sustains the below-ground ecosystem. Those essential features were present in the first forests, and they set the trajectory that forest ecosystems have followed ever since. The discovery of Devonian fossil forests in Morocco, dating to approximately 372 million years ago and preserving multiple species of early trees in a single site, has provided new information about the composition of North African forests near the end of the Devonian period. The Moroccan site, which was described in the scientific literature in 2024, contains a diverse assemblage of tree species, including both cladoxylopsids and early seed ferns, suggesting that by the late Devonian, forests were composed of multiple species coexisting in the same landscape rather than single-species stands. The presence of different tree architectures in the same fossil site, some with the bundle wood structure of the cladoxylopsids and others with the more modern solid wood structure of the seed ferns, provides direct evidence for the ecological diversity of late Devonian forests that was not previously available from any other site. Each new discovery of a Devonian fossil forest adds new data points to the picture of how forests evolved and spread, and the picture continues to be revised and enriched with each field season. The global deforestation of the modern era is reversing, in compressed and accelerated form, some of the ecological processes that the first forests initiated 385 million years ago. When forests are cleared, the deep organic soils that developed under centuries of forest cover are exposed to erosion, and the nutrients they contain are rapidly lost. The mycorrhizal networks that connected forest trees are destroyed. The carbon stored in the soil organic matter is released to the atmosphere, and the local climate becomes warmer and drier as the moderating effect of the forest canopy on temperature and humidity is eliminated. Those processes are essentially the same as the processes that occur when forests are eliminated by natural events such as volcanic eruptions or extended droughts, and they demonstrate that the ecological services provided by forests are not permanent features of the landscape but are maintained continuously by the biological processes of the living forest. The first forests created those services through hundreds of millions of years of evolution. Modern deforestation can destroy them in years. The relationship between forests and water, which is fundamental to the functioning of modern terrestrial ecosystems, was established by the first Devonian forests in ways that are still operating today. Forests do not simply receive precipitation and release it as runoff. They actively modify the hydrological cycle, evaporating large quantities of water from leaf surfaces through the process of transpiration, which returns water vapor to the atmosphere and contributes to the formation of clouds and precipitation downwind of the forest. The Amazon rainforest, which transpires enormous quantities of water into the atmosphere and is estimated to generate a significant fraction of its own rainfall through that process, is the modern expression of the relationship between forests and water that began with the first Devonian trees. The Devonian forests of the tropical latitudes of the time would have had the same effect on the local water cycle, creating a self-sustaining feedback between forest cover and rainfall that helped to maintain the humid conditions that the forests required. The sounds of the Devonian forest, if it is possible to imagine them, would have been fundamentally different from the sounds of any modern forest. The absence of bird calls, which are the most prominent biological sounds in most modern forests, would have been total. Birds did not appear until the Jurassic period, approximately 170 million years after the first forests. The absence of mammalian sounds, from the rustle of small rodents in the leaf litter to the calls of larger mammals, would have been equally complete. Mammals did not appear until the Triassic. The dominant biological sounds would have been the buzzing and clicking of the first land arthropods, and the occasional splash of an early tetrapod moving through the shallow water at the forest margin. The wind in the canopy would have sounded recognizable, as the mechanical interaction of wind and leaves does not require any particular evolutionary history. But the overall soundscape would have been sparse and strange by modern standards, a quiet biological world in which the chemistry and the ecology were active and transformative, but the acoustic dimension of life was only beginning to develop. The evolutionary arms race between plants and the organisms that eat them, which the first Devonian forests set in motion when they created the first significant quantities of plant biomass on land, is one of the most productive sources of biological diversity in the history of life. Every innovation in plant defense, whether chemical, structural, or phenological, has been followed by counter-adaptations in the herbivores and pathogens that exploit plants, and every counter-adaptation has driven further innovation in plant defenses. The extraordinary chemical diversity of plants, which includes tens of thousands of secondary metabolites found nowhere else in the living world, is the product of that arms race, and it is the source of most of the pharmaceuticals that modern medicine depends on. The aspirin derived from willow bark, the quinine derived from cinchona bark, the morphine derived from the opium poppy, and the Taxol derived from the Pacific yew are all products of plant chemical defenses that evolved over hundreds of millions of years in response to the selective pressure of herbivores and pathogens. The first forests initiated that arms race by creating the first substantial quantity of plant biomass on land. The philosophical dimension of the story of the first forests is one of the most quietly profound in the history of science. The trees did not know they were changing the world. They had no awareness, no intention, no purpose beyond the immediate biological imperatives of growth and reproduction. And yet the cumulative effect of their growth and reproduction, multiplied across millions of individuals over millions of years, was a transformation of the global environment so thorough that the world before and after their appearance is barely recognizable as the same planet. That gap between the intention of individual organisms and the consequences of their collective action is one of the most important insights that the history of life can offer, that the most consequential changes in the history of the Earth have been produced not by deliberate acts but by the unintended collective effects of organisms following their biological nature. The first forests changed the world by accident. So, in a different way are we. The international research community working on the first forests is a small but productive group of scientists drawn from geology, paleobotany, geochemistry, atmospheric science, and evolutionary biology, and their collaboration across disciplinary boundaries has been essential for the development of the integrated understanding of the Devonian forest revolution that exists today. The paleobotanist who describes a new fossil tree species depends on the geochemist to date the rocks in which it was found and to reconstruct the atmospheric conditions under which it grew. The atmospheric scientist who models the carbon dioxide drawdown of the first forests depends on the paleobotanist to provide the biological parameters, the leaf areas, the root depths, and the wood accumulation rates that feed the models. And the evolutionary biologist who reconstructs the phylogenetic relationships of the first tree lineages depends on the paleontologist to provide the morphological data from the fossils that constrain those relationships. The story of the first forests is a genuinely interdisciplinary story, and understanding it fully requires the integration of many different kinds of evidence and expertise. The living descendants of the first forest ecosystems surround most of us every day without our recognizing their ancient heritage. The ferns in a garden are the modern representatives of the lineage that produced watiza and the other early trees. The horsetails growing in a damp ditch are the modern representatives of the lineage that produced the giant calamites of the Carboniferous coal forests. The ginkgo tree in a park, with its distinctive fan-shaped leaves, is the last surviving member of a gymnosperm lineage that was already ancient when the dinosaurs appeared. And the conifers that make up the boreal forests of the northern hemisphere are the modern representatives of the gymnosperm lineage that replaced the coal forests at the end of the Carboniferous. Those living plants are not living fossils in the sense of being unchanged since the Devonian. They have evolved continuously and substantially since their lineages diverged from the first forest trees. But they are the living endpoints of evolutionary trajectories that began in the Devonian, and they carry in their biology the deep heritage of the first forests that made the terrestrial world habitable. The mystery forest that existed before trees is not really a mystery anymore. We know its names, Watiza and Archaeopteris, and the 100 other species that populated the first forests of the Devonian. We know how they worked, the bundle wood of the Cladoxylopsids, the true wood of the progymnosperms, the spore-bearing structures of the Devonian ferns. We know what they did to the world, drawing down carbon dioxide, building soil, changing rivers, fertilizing oceans, triggering extinctions. And we know what they started. The 385 million-year experiment in terrestrial life that continues to this moment, in every leaf and root and mycorrhizal network in every forest on Earth. What was mysterious was not the forest itself, but our ignorance of it, an ignorance that paleobotanists have been steadily reducing for more than a century. The mystery is mostly solved. What remains is the wonder. The first forests also transformed the visual character of the Earth's surface in ways that would have been apparent from space. Before forests, the continents of the Earth were predominantly gray and brown, the colors of bare rock and mineral soil, with patches of green where the earliest land plants had colonized the most hospitable environments. The spread of the first forests covered large areas of the tropical and subtropical continents with a continuous green canopy, changing the reflectivity of the land surface and the absorption of solar radiation. That change in the Earth's albedo, the fraction of incoming solar radiation that is reflected back to space, had small but measurable effects on the global energy balance, contributing to the cooling that accompanied the carbon dioxide drawdown of the first forests. From space, the greening of the Devonian continents by the first forests would have been one of the most dramatic visible changes in the appearance of the Earth since the colonization of the land by the first simple plants in the Ordovician. The first forests were, in the end, the most consequential biological innovation in the history of the terrestrial world, and their appearance marked the beginning of the modern era of terrestrial ecology in a way that no subsequent event has equaled. Everything that followed, the coal forests of the Carboniferous, the conifer forests of the Mesozoic, the flowering plant forests of the Cenozoic, the human civilizations that depended on forest timber, agriculture, and fossil fuel, can be traced back in a continuous line of ecological consequence to the moment when Watiza rose above the low-growing vegetation of the Devonian landscape and cast the first shade. That shade, however small and temporary, was the beginning of a different world, a world with deep soil, high oxygen, stored carbon, diverse terrestrial fauna, and the biological complexity that eventually made intelligence possible. The first trees made the world we know. They did it by growing tall, by dying slowly, and by changing everything without intending to. If this documentary kept you company tonight, subscribe to the channel. There are more worlds to explore, more geological and biological revolutions to trace, more moments when life changed everything by accident, more events in the deep past that are still shaping the present moment. A new one every week, always in the dark, always going further than you expect, always finding that the world is stranger and older and more consequential than any single documentary can capture.